live in a global pandemic, and I'm sure all of us are thinking about viruses a lot these days. For me, I think about viruses all the time. When I think about a virus, I don't necessarily see an infectious agent. All I really see is a tool, a platform technology that we can engineer to impart new functionalities targeting human health. And the viruses that I think about and that I consider materials are plant viruses. So my early training was in plant virology, where I learned how to work with plant viruses and become to appreciate them as tools for bioengineering and materials research. Before I get started, I want to declare conflict of interest. I'm also the co-founder and serve as the Chief Scientific Officer of Mosaic Immuno-Engineering. So viruses as nanotechnology or viruses as materials. My lab works in the space of nanomedicine. And as nanoengineers, we seek to devise nanoscale objects, nanoscale machines that can interface with the body, that can deliver a payload to specific cells and tissues. And viruses naturally perform these functions. Viruses are evolved to package and encapsulate a cargo, and they're programmed to deliver this cargo to specific cells and tissues. And in our lab, rather than building these sophisticated nanoscale machines from scratch, we have turned towards nature's materials, namely the plant viruses, and are repurposing them for the desired applications. I think there's at least 1,001 good reasons why we've chosen a plant virus as a nanotechnology platform. As I just said, these can be regarded as prefabricated, naturally occurring nanomaterials, naturally expert at the delivery of cargo. They are non-infectious towards mammals. So compared to the mammalian viral vectors that are already explored in medicine, the plant virus offers another layer of safety. Compared to the synthetic materials, we have a very high precision nanomaterial, monodispersed and uniform. Each particle, to a near approximation, is a clone of each other. They are identical. We have a high degree of symmetry and polyvalency. Many structures are known to atomic resolution, and not only are they known to atomic resolution, we can engineer and tailor them with atomic precision. The platform is scalable and programmable, and we produce these plant virus-based nanoparticles in plants. So going from black-eyed peas to nanotechnology. So we use two general methods to initiate the infection or the expression process. One is termed mechanical inoculation. This is used to produce live virus particles, infectious towards plants, yet non-infectious towards mammals. Here we take purified particles and we gently rub them onto a leaf to induce an infection in the plant. About 2 weeks after infecting the plants, the leaves, this is a cowpea plant, the leaves will turn from this uniform green coloration to these mosaic symptoms, indicating the plant was successfully infected. So we're making use of biology to make copies of nanoparticles. We can then harvest these particles using straightforward methods. We blend the leaf material using a household blender. This then, smoothie then undergoes multiple steps of centrifugation to extract purified virus particles. We can also make use of a technique called agroinfiltration to produce VLPs, virus-like particles. So these are materials that are devoid of the nucleic acid, so meaning that they are non-infectious also towards plants. The yields depend on the specific particle to be produced, but they can be up to 500 milligrams of virus particles from just 100 grams of leaf material. 100 grams of leaf material is a medium-sized Ziploc bag. You can extract 100 to 500 milligrams of virus particles. We use these viruses, these virus nanotechnologies to treat cancer, and we've begun the treatment of canine patients, so pets diagnosed with cancer. And these patients are dosed with only 1 milligram of virus particles. So just 30 to 40 plants giving us 100 grams of leaf material could treat up to 500 patients. A few words about the chemistry and the designer engineering design space that these platforms give us. Here's the structure of the tobacco mosaic virus. Tobacco mosaic virus forms a nanotube. These are 300 nanometer in length, 18 nanometer in width, with a 4 nanometer central channel. The inside and the outside is chemically distinct. So this means if we wanted to formulate a drug carrier, we could place a therapeutic cargo on the inside of the particle, making use of carboxylate specific chemistry, so targeting glutamic acid side chains shown in green. So we can covalently link a therapeutic payload to the interior of the particle, and then use amine functional handles on the exterior surface to introduce, to enhance biocompatibility by conjugating polymers, or by inducing tissue specificity, by incorporating a targeting ligand, a peptide, an antibody fragment, for example. We can also deliver therapeutic nucleic acids, shown in red as the natural cargo of the particle, in a single-stranded RNA genome. And we can replace that with any nucleic acid sequence of interest. So we have a platform that can carry a cargo that can be engineered to gain tissue specificity and to target specific molecular features within the body. When it comes to the application areas, my lab works on a number of applications. And first, the molecular imaging to aid risk stratification. Here we're focusing on nanoparticles, plant virus-based nanoparticles carrying gadolinium, molecules such as gadolinium, enabling MRI imaging. We are working in the space of drug delivery, where the cargoes or the payloads of interest are either chemotherapy, protein drugs, or nucleic acid therapeutics. More recently, we've begun to apply the concepts of nanomedicine to agricultural nanotechnology. So engineering plant viruses to deliver pesticides to specific regions of a plant or deep into the soil. And lastly, we work on immunotherapy and vaccines. And that's the area that I will highlight here today. So in some aspects, we think about these virus particles as a way to deliver a cargo. And that's what we're exploring when it comes to molecular imaging, delivery of therapeutics, or delivery of pesticides. But the virus particle itself can function as a therapeutic. And that's what I'm going to explain in the next part of this talk, highlighting two applications. One is an immuno-oncology application using a plant virus-based therapy to treat tumors. And the second is the story how we pivoted the technology towards applications as a COVID-19 vaccine candidate. So to put this into a context, I first want to explain the cancer immunity cycle. The cancer immunity cycle is our body's own defense against a tumor. And this is explained in this cartoon here. So in step one, a tumor cell will undergo apoptosis. And this will release, therefore the cell will release its contents into the microenvironment, into the tissue. So tumor-associated antigens are being released. Within the tumor microenvironment, within our tissues, we have innate immune cells programmed to sample the contents of dying cells. And these cells will realize there's something not quite right here. They will recognize there are these tumor-associated signatures. This could be a protein overexpressed. This could be a mutated protein. So these innate cells now process these tumor-associated antigens. They travel to the lymph nodes, and that's when they start talking to the adaptive arm of the immune system. So they start engaging with cytotoxic T cells, and they will program these cells to say, when you find this signature, this tumor-associated antigen, then engage with the cell and kill the cell. And that brings us to step 7, where we see a T cell engaging with a tumor cell, inducing apoptosis, which leads to more release of tumor-associated antigen, and the cycle continues. So this is our body's own defense against tumors. However, over time, the tumor will shut down this cancer immunity cycle by a number of mechanisms. Cells can no longer enter the tumor bed. Cells within the tumor microenvironment will be reprogrammed from tumor fighters to become tumor supporters. So we end up with an immune suppressive state within the tumor. So what we thought to do is to use a virus, a plant virus nanotechnology, and to specifically use it as an in situ vaccine. So here we're taking the plant virus nanoparticle and we're injecting it directly into an identified tumor. The idea is, even though the plant virus is non-infectious towards mammals, the plant virus still has features to be recognized by the immune system. Specifically, plant viruses are recognized by toll-like receptors, so pattern recognition receptors. These recognition events will essentially trigger alarms, like an alarm system to the immune system. So the immune system is fooled into thinking there is an infection, yet there is not. However, as part of this process, innate immune cells will be recruited and rushed to the tumor. Within the tumor microenvironment, because the virus signals through these toll-like receptors, there's chemokines and cytokines that will stimulate immune cells. So we're going from immune suppressed state into an immune activated state. And this process eventually relaunches the cancer immunity cycle. And I will now show you a series of data to show that this indeed works. So by this in situ vaccination approach using the plant virus nanoparticle, we've demonstrated potent efficacy in a number of tumor mouse models, including melanoma, breast cancer, colon cancer, ovarian cancer, and glioma. It is very important to understand that even though we're treating locally, the response that we are getting is a systemic anti-tumor immune response. Here's some data, for example, from a treatment of glioma in mice. So what you're looking at is an MRI scan of a mouse brain. Within the brain and the PBS control group, you see a large mass. That's a tumor, a glioma, orthotopically grown in the brain of a mouse. After 2 treatments of intratumoral injection, the survivors are essentially tumor-free, as shown on the right, CPMV. CPMV stands for cowpea mosaic virus, and which is the plant virus nanoparticle that is being used here. We're inducing not only systemic effects, meaning if we have a primary tumor and metastatic disease, the therapy will act on both the primary tumor that is injected and the distant metastatic site. And we're inducing immune memory. So here's a data set where we treated a dermal melanoma. You can see in panel A, a mouse bearing a dermal melanoma. The tumor on the left was injected with the virus nanoparticle, CPMV. The image was taken 3 days after, and the tumor essentially is collapsed. So that made us wonder, is there any toxicity of the particle itself? And the answer is no. If we take tumor cells and the virus particles and just incubate them in a petri dish, there's no effect of the virus particle onto the tumor cells. However, if you take tumor cells and innate immune cells, say macrophages, and you then add the virus particles, then the virus particle will act on the innate cells, on the macrophages, to activate them to attack the tumor cells. And that's what we're seeing in this image here. This tumor necrosis is not due to the particle having any toxic action on the tumor cells, but rather the particle is just activating the innate immune cells to do their job, to kill the tumor. We see about 50% survival with this particular dosing and treatment regimen. If we then take the survivors and we re-challenge them with the same tumors, the tumors essentially don't take. So that's shown in panel D. The re-challenged mice, we have only 1 mouse with a small tumor, several mice, there was no tumor growth. So this indicates that we have engagement of the adaptive immune system and we're inducing immune memory. So mechanism is summarized in this slide. Essentially, the key point is that the CPMV plant virus-based in situ vaccination essentially functions as a switch to turn a cold, immune-suppressed tumor into an immunological active hot tumor, We are remodeling the tumor microenvironment to relaunch the body's defense against the tumor. We've moved this beyond the treatment of mice and are currently treating canine patients. I should also mention the collaborators here. So all the work I just presented is done in collaboration with Stephen Firing at Dartmouth College. Then the treatment of companion dogs is done in collaboration with Steve as well as Jack Hoops. Both are located at Dartmouth College. So here we're looking at a patient, a canine patient, somebody's pet that was diagnosed with melanoma. This patient came in with a large tumor, 5 centimeter large tumor. The patient received a combination of radiation and the plant virus-based therapy. The combination is given because the radiation is standard of care. It's somebody's pet, so we don't want to withhold standard of care. 2 weeks after treatment, we already see dramatic reduction in tumor burden. But what is important that after 6 months, there's no detectable tumor, there are no detectable metastatic sites. We have treated about 20 canine patients to date, all responded to the treatment with no apparent side effects. Many are still alive. All of them were alive 10 months post-treatment. One dog was alive 3 years after treatment. When these patients receive radiation only, their life expectancy is much shorter than that, on the order of 6 to 9 months. So we have a novel immuno-oncology therapy, a therapeutic candidate, and we're eager to move this out of the lab into a human clinical trial. So switching gears just for a couple of minutes. So last March hit, and all of us, right, as scientists, engineers around the world, started thinking about COVID-19 and what contribution we could make toward ending this pandemic. And my lab started thinking about COVID-19 vaccine candidates. And the challenges with any vaccine is always the vaccine design itself, the choice of the adjuvant, the choice of the antigen, the choice of the delivery system, but also manufacturing global distribution. And the rollout of the current vaccines highlights the difficulties with vaccines that need to be stored at ultra low temperatures. The cold chain requirements bear logistical and fiscal barriers, not only in the resource poor areas of the world, but even within the US. So we started thinking about this and thought the plant virus nanotechnology may offer a unique handle for vaccine development. I just showed you CPMV acts as a very potent adjuvant. It has an ability to stimulate the immune system. So per definition, that makes it an adjuvant. But we can also use it as a display technology. And I go into this in this slide. So what I mean by display technology is that we can use the plant virus-based nanotechnology and engineer it, modify it to display epitopes. So these are antigenic sequences from the SARS-CoV-2 virus. So we can produce a chimera that has, per plug and play chemical biology approaches, can display these antigenic epitopes on its surface. So the core is a plant virus with 2 functions: to deliver these epitopes, to stabilize them, and to activate the immune system. So the plant virus is our adjuvant and delivery platform. And the displayed peptides, these are our epitopes to stimulate the immune response and to generate a SARS-CoV-2 specific immune response. Our plant virus nanoparticles can be stored at room temperature, so we have the potential to overcome any cold chain requirements. Here's some preliminary data. So we generated a number of SARS-CoV-2 vaccine candidates, and we tested their immunogenicity and potential to elicit protective immunity using mice. So healthy mice were immunized with our vaccine candidates based on the cowpea mosaic virus. And what you're looking at in the slide is antibody titers against the S protein, the spike protein. So that's our target. And we're showing that all our vaccine candidates elicit potent antibody responses that bind the S protein. We've more recently shown that 5 of these candidates are indeed neutralizing SARS-CoV-2. And this was done in the BSL-3 labs at NIAID. So we have vaccine candidates that indeed are neutralizing in an in vitro assay SARS-CoV-2. So this is very exciting, but we want to bring this one step further. And what we want to do, and we're doing this as we speak, we're developing a microneedle patch technology. The plant virus nanotechnology is extremely stable to temperature. So has a high degree of temperature ranges that you can expose these particles to, and they will maintain their structural integrity. So presently we're using hot melt extrusion and injection molding as a technique to produce microneedle patches. The advantage of using the advanced manufacturing, hot melt processing and injection molding is that these techniques are massively scalable. And this would not be possible with many other biologics or with other nanoparticles, because this is done at very high temperature. So the process is done at 90 degrees C. But the plant virus, when in the dry state, is extremely stable and can maintain these high temperatures. So we're using these processes to make these microneedle patches. Our technology includes a slow release technology, and this, if effective, would also provide an effective shield to the patient after a single administration. So we are alleviating the need to see the doctor's office and to see them twice. So we're alleviating the need for repeat administration, and we are also generating a platform that can be shipped without the cold chain and self-applied. So to us, this is a very exciting time to pivot our technology going from immuno-oncology to infectious disease. And I want to point out also the collaborator. This work is done in collaboration with John Pokorski, who is developing the advanced manufacturing techniques. Yeah, with this, in the interest of time, I will come to an end. We are exploring plant viruses as a nanotechnology platform for many different applications. I highlighted today the immunotherapy and vaccine applications. This is my lab. Obviously, this picture was taken pre-COVID. And yeah, I want to thank all my lab members, the collaborators, as well as our funding sources. Thank you very much.